Alan, thanks for having us over this week to the Small Scale Bioprocess Development Lab. So it seems to me, and we're looking at a couple of these bioreactors here now, it takes a lot of experience and expertise to be able to put one of these things together. It seems very complex to me. Maybe you could tell us a little bit about what we're looking at here.
There's a lot of parts and a lot of components. I think the most important one for me is probably the agitator motor, which sits on top and spins really hard to make sure that everything is well mixed. Underneath that, I've got my heating blanket, which makes sure the cells are at the right temperature. And then I've got a lot of ports, which I can connect to other things.
Yeah, this looks like an octopus. I can see all kinds of things coming off of there. What are all these tubes for?
Some of them are going to things like nutritional complements. So like this is our feed media. We can deliver more glucose or amino acids and things like that to the cells. We have another piece of tubing that connects to a bottle of base so that we can control pH by adding something that's basic. And we also have ways that we can deliver oxygen. So coming down the top, we've got a line that can deliver oxygen so we can make sure the cells have plenty of oxygen. We work for typically about 2 weeks with each production culture in order to get the most protein we possibly can.
2 weeks, and so that's really where all of this comes in then, is the ability to add media to it over time so that they can be fed properly. And how do you start to control those elements?
So to control things, as you can see maybe in the back, we have one probe that's measuring dissolved oxygen and one probe in front of that measures pH. That helps us to monitor things online to be able to respond as needed.
So the computer sees what's happening in the culture. It has some set point that you've decided upon probably, and then the system is adding base or media as needed into the system.
Yes, so both base and nutrients can be delivered through the peristaltic pumps, which just run around in a circle and eventually apply pressure such that things are pushed inside the bioreactor.
And so a lot of what we're looking at then is really about monitoring what's happening in the system and then providing a response to that to be able to provide the right nutrients or oxygen.
That's exactly the way it is.
Where do you learn how to put one of these systems together?
Well, just right in front of them. You just have to pick up
You just
the
have to
pieces,
work on it. Well, let's go put one together then.
Absolutely. We've got ones right over here.
Excellent.
So this is kind of the metal skeleton of everything. So it's all the stainless steel parts. We've got a base support, which everything goes on, and then lots of positions to put inputs.
So for different gases or probes, or maybe we bring the media in.
Yeah, so that's why we've got triport here. So lets you put in 3 different things all at the same time. And there's actually another position that you can have another triport there. You can bring up to 6 things all at the same time if you needed to. So we actually have 2 different impellers here. So we've got one marine and one Rushton impeller, and you can see that there's different blade types. We've done computational fluid dynamics to show us this gives us the best mixing possible.
And this is the sparger?
This is the sparger as well, yes.
And how do you make decisions about which sparger you use?
So there's 2 different sparge types that we use most often. One is called a drilled hole sparger, and one is a sintered sparger. The main differences are bubble size, and therefore how much mass transfer you get between oxygen and then stripping of carbon dioxide as well. So here we have a drilled hole sparger, and you've got very small holes, but you can still see them as individual
just barely,
holes.
But they are there, yes.
If you switch to a sintered sparger, you can't even see the individual holes.
Oh, it's almost like a filter.
It's basically is a filter for oxygen that's basically coming in with very, very tiny bubbles.
So you might run calculations to try to understand how much oxygen is being distributed, as well as how that might impact overall aspects of your process.
That's exactly what the computational fluid dynamics is all about, is making sure that we can just deliver the oxygen we need to, and also remove the carbon dioxide to keep it from poisoning the cells.
And at this stage in process development, you might test a few different parameters to see which one works the best.
That's exactly the way we do it. That's why we have the benches of 8. So the glass vessel comes next, and that's this container here. And then the next piece is to make sure that we don't lose all of the liquid. So when we sparge oxygen through, we're actually going to be carrying liquid out, and that's what this condenser is all about. It's got 2 pieces. Unscrew on the top. You're actually going to get liquid that's going to condense at each one of these stages.
On a column.
Exactly. This is the same thing as plates on a column, and there are several of them. And that result is that the liquid will condense and fall back into the bioreactor, maintaining the liquid volume in your bioreactor.
Since we're heating the system and we're blowing gas through it, it's going to come up through there. So does this sit on the top of the system then?
It does. You screw it all together. And then you can attach it to the top of the bolt.
Excellent. So you would put all of these pieces together, and then it seems like the only thing missing is the hosing that we saw over on the other system.
Yes. So I have right over here another completely contained container.
Excellent.